Yes.
Okay. So good morning, everyone. My name is Victor, and I'll present part of my thesis work on the role of habitat complexity on benthic faunal diversity of subtidal maerl beds. The project took place at the UM and was supervised by Jardim and Olivier Gauthier. And first, I would like to introduce you to the topic. So the role of habitat complexity on biodiversity remains one of the fundamental questions in ecology since it was first brought up by the MacArthur brothers in the 60s. They showed that bird diversity increased with habitat complexity, and they used vegetation high diversity as a proxy for it. Since then, several studies have been conducted on the topic, and a cornerstone of ecology is that complexity promotes species richness and abundance by providing more niches and improving available space. However, the generalization of the topic remains constrained by methodological problems. For example, studies usually relied on simple correlations and most fail to include possible confounding factors. However, we point out that identifying the isolated effect of habitat complexity on biodiversity can be quite complex as human impacts and environmental conditions can both affect biodiversity directly or indirectly through habitat complexity. And additionally, species compositions and abundance by themselves can also change the habitat complexity and the physical environment. So not surprisingly, the topic became especially popular in the aquatic systems as they are faced with important physical and biological pressures, such as currents and intense predation. In those systems, the 3D complex structures are thought to be a crucial source of shelter. And most of the studies in the marine realm have focused on biogenic habitats and are based on the effect of a single foundation species, which are key ecosystem engineers that are spatially dominant and improve available space. For instance, corals and macrophytes. However, most of these studies were qualitative, usually in comparing communities where the foundation species is present to communities where it's not, and the effect of complexity has been often addressed as an a posteriori explanation for observed patterns instead of being directly assessed. Additionally, even when studies quantified elements of habitat complexity, only a few of them evaluated their effects on diversity as their main hypothesis. And this question has been explored in many marine environments, but maerl beds are ideal candidates to investigate it. And that's because they are ecologically important biogenic habitats that are founded on a few species of free-living coralline algae. And their high biodiversity has been long justified by the high 3D complexity that those algae provide, as we can see here. However, very few attempts to quantify this complexity have been made. And in Brittany, in France, maerl beds are present all along the coast and therefore occur in different environmental conditions, which makes the area very attractive for exploring the topic. And that is exactly what I've been working on during my thesis. So our main objective was to investigate the role of habitat complexity on diversity by studying the links between the complexity provided by maerl beds, the physical environmental conditions that they are exposed to, and the benthic macrofaunal communities that they harbor. Our specific goals were to understand regional patterns of maerl habitat complexity and its effects on the local faunal diversity, on community stability, and finally, on the regional diversity patterns. But I'll present our main findings only on the first three topics. So in order to explore all those questions, we used data from the long-term monitoring program, Rebond. 6 years of macrofaunal and sedimentological data from 10 maerl beds have been analyzed, and every year each bed was sampled in 3 different fixed points, and 3 replicates were taken with a van being grabbed in each point. Additional maerl thalli were sampled during the last 2 campaigns, and for each point in each bed, I quantified the complexity of 12 maerl thalli, totalizing 360. Additionally, hydrological data for the 6 years period and bathymetry were also acquired through regional models. The complexity was quantified through common thalli-specific morphometrics that have been previously suggested as important to the associated fauna. First, the mean benching density was measured by randomly placing a 1 centimeter quadrat on top of the algae 5 times and counting the apical branches that are contained in it. Then, the largest diameters of the 3 main axes of each thalli were measured 3 times with a vernier caliper. The diameters of the largest, intermediate, and smaller diameters were then used to calculate sphericity and 2 other diameter ratios that describe rhodolith shape from discoidal to ellipsoidal and spheroidal. Additionally, Since the macrofauna was sampled at the habitat level and not at the maërl level, we also analyzed available data on the maerl density in the study beds. However, this data was available only at the site level and not at the point level. We also conducted fractal analysis, which is a method that has become popular in ecology because it is a measure of complexity ideal to describe intricate patterns and shapes, like those of many organisms. It's also independent of the studied object size and therefore is a very comparable metric. The fractal dimension is in practice a measure of complexity as a change in detail with change in scale, as it counts the number of repeated patterns in different scales of an object. In order to quantify it, I photographed the 360 tally using a macro lens and applied a focus stack in order to keep part of the algae's 3D complexity. Then, the final pictures were transformed in binary and grayscale images to be analyzed using Fractal, which is an open source plugin for ImageJ. And binary analysis gives us information on the complexity of the tally's outline, while grayscale analysis keeps the textural information. So going back to our first question, we wanted to investigate how complexity varies along the 10 study sites. And because numerous studies relate maerl morphology to growth patterns to environmental constraints, we expected that maerl complexity would vary at least partially due to environmental conditions. So to test this, multivariate analysis was performed, and for this and the following analysis, the median complexity value for each sampling point was taken. Two ordination methods were performed, a principal component analysis, or PCA, of the complexity metrics, as well as a redundancy analysis, or RDA, of the complexity in function of the environmental factors. Additionally, each complexity variable was modeled at a time in function of all environmental variables in order to further elucidate some of the patterns. So the PCA showed us that while some complexity metrics were redundant, most were very complementary. And the first two axes represent around 64% of the variation among the beds. The first axis was mainly driven by the smaller tally diameter and the two fractal dimensions that we measured. While the second axis was most driven by the maerl density, the branching density, the sphericity measures, and the largest tally diameter. The PCA also showed us that there was a broad range of habitat complexity levels among the beds in Brittany, with 4 main groups of complexity that are also supported by cluster analysis. So the first group contains the 2 sites at the Bay of Brest, which are Rosga and Caralieu, and they present some of the highest maerl densities and small but highly branched, spherical, and complex tally, and therefore are considered the most complex habitats in the study. Then, the second group is composed of 5 beds which present intermediate to low branching densities, sphericities, complexities, and maerl densities. And then the third group contains all the 3 points of the Trévignon bed and one of the points from the Molène bed, all characterized by low maerl densities but containing big and highly branched spherical and complex tally. And finally, the last group is composed of the Saint-Brieuc bed and the remaining points of Molène, which contain the biggest maerl tally but have the lowest maerl densities and low values of sphericity, branching density, and fractal dimension, being considered the least complex habitats. So the redundancy analysis of complexity in function of environmental variables was not significant. However, the different aspects of complexity may be driven by different processes, and those relationships can be very intricate. Therefore, looking at all of the factors at once would probably require more samples. So we decided to model each complexity variable at a time in function of all environmental variables, and the final selected models showed us a few things. So first, sphericity decreased with current velocity, which may indicate that maerl growth is more homogeneous under slower currents, allowing all faces of the maerl tally to receive light. We also saw that maerl density decreased with depth, which is a pattern that has already been described in the literature and that might be related to light availability as well. So now our second question was whether or not all those complexity metrics were relevant to local diversity. And based on extensive literature for several environments, we expected that local macrofaunal diversity would increase with habitat complexity. In order to test this, species richness, total density, Simpson's inverse, and diversity index, and p elusiveness were all estimated for each sampling point. Then, Each diversity metric was modeled against the axis of the PCA, and since no metric of complexity alone was sufficient to totally describe the habitat complexity. These also allowed more parsimonious models. So the two selected linear mixed models in which site was the random factor showed us that the variables in the second axis of the PCA were relevant to local diversity. The first model showed us that the combined effects of maerl density, branching density, and sphericity promote species richness, and that the beds with the biggest tally were the ones with the lowest richness. This model had an R squared of 0.27 for the fixed effects only, and a total R squared of 0.36, which means that 9% of the variation explained by the model was related to site variations. So then we included the environmental factors of each site, and we could see here that the percentage of variance explained by the model due to other non-measured features was very reduced to only 2%. And we can see that depth has also a negative effect directly on species richness, which highlights the importance of a holistic approach. So previous studies have shown that, moving on to our last question, sorry. Previous studies have shown us that faunal communities from biogenic habitats are more stable than those from bare sediments. And this study from Boyer and collaborators developed in the same study region as the present study showed that maerl communities were the most stable of 4 habitats, as we can see here. And the authors hypothesized that this was due to their habitat complexity. Our goal was then to test if this hypothesis would hold within the maerl beds only. And to test it, we measured the temporal beta diversity for each site as the total variance of the community data during the 6 years selected. Those values of beta diversity were then modeled in function of the axis of the complexity PCA. So we found that the relationship between habitat complexity and community stability was intricate, and that linear approaches weren't adequate for describing them well. Indeed, the points where the temporal diversity were the highest, and therefore the communities were less stable, were the ones with lower habitat complexity, as we can see here. However, in the sites with intermediate complexity, temporal diversity increased again. So we argued that there could be several confounding factors, such as environmental conditions that we didn't measure, like exposure, for example, and the histories of the beds. Additionally, the fauna on its own can influence stability. For example, the Trevignan site has very low maerl density, and therefore we expected it to be less stable, but it's highly dominated by the porcelain crab Cydia longicornis, in a way that its temporal diversity is quite low, as you can see here in the purple points.
2 minutes, 2 minutes.
So in this study, which to our knowledge is the first one of its kind in maerl beds, we were able to demonstrate that there were clear differences in habitat complexity between some of the 10 beds analyzed. And we saw that those differences could be related to both hydrodynamics and bathymetry, and demonstrated that depth has a direct effect on both complexity and richness. We also saw that only a few of the measured complexity variables were relevant to the fauna, with special attention given to maerl density and branching density. And we also saw that richness was higher in the more complex beds. Finally, we started exploring how habitat complexity affects stability, and although further research should be done on this topic, there seems like complexity does play a role in it. However, I would also like to point out that the study has several limitations. For one, we expected that the fractal dimension would be important for macrofaunal diversity, and that puts the adequacy of our estimation into question. Although it might be that indeed those measures are not important at the scale of the maerl tally. And secondly, our environmental data set was contained only a few constraints, and other aspects such as exposure might be important. Another limitation was the use of linear statistical methods, as we've seen that in many cases the relationships are quite intricate. And finally, we highlight 2 main limitations which should be taken into account in future studies. We believe that looking into the endo-epifaunal compartments separately could yield very different results, and that a taxonomic approach might not have been the most adequate one since at the core of habitat complexity theory is the hypothesis that complexity provides more niches, and therefore using a trait-based approach could be more interesting. So that was it. Thank you very much for your attention. I'll gladly answer any of your questions